DNA separation by gel electrophoresis. That is actually for separation of DNA, we're using agar gel electrophoresis. Agar gel electrophoresis is the easiest and most popular way for separating and analyzing DNA. Here, DNA molecules are separated on the basis of charge by applying an electric field to the electrophoretic apparatus. Shorter molecules migrate more easily and move faster than the longer molecules, that is the heavyweight molecules through the pores. That is, these pores are actually in the agarose molecules, and these pores are actually in the gel. Uh, and this gel was formed with the use of the agarose, and this process of movement of the molecules through these pores is called sieving. The gel might be used to look at the DNA in order to quantify it or to isolate a particular band. The DNA can be visualized in the gel by the addition of some staining agents such as ethidium bromide. So, for instance, if we are asked, so why to run a gel, and we have to know that molecular weight markers and DNA fragments of known size are used along with the desirable bands we have to identify, and in this way, along with the desirable bands, along with the products, we have to run this type of the known bands for these purposes, and comparison of sample bands to the markers allowed visible confirmation of the desired products which have to be desired from the experiment, and if it is got or not, we can easily justify, we can come to know by using this type of reference bands which are used. And quantification of sample DNA is also can be helpful from these desirable bands, as these brands have different quantification reference bands, and comparing with this, we can easily able to know approximately what amount of DNA is there in that specific band. Come to the application forms part. This gel electrophoresis is mainly used in different molecular biology laboratories, different forensic studies, as where the DNA samples and DNA related experiments are going on. Estimation of the size of DNA molecules following restriction enzyme digestion. For example, in restriction mapping of clone DNA, this that is agar gel electrophoresis is used, and through this process only we can easily come to know that this restriction enzyme has able to work or not. Similarly, analysis of PCR product, that is in molecular genetic diagnosis or genetic fingerprinting, separation of restriction genomic DNA prior to southern transfer or of DNA prior to northern transfer. For this type of analysis also, we have to use the gel electrophoresis, that is agar gel electrophoresis. And biomolecules are separated by applying an electric field to move the charged molecules through an agarose matrix, and the biomolecules are separated by size in the agarose gel matrix. So it is a very useful method which is widely used in different fields of molecular biology.